Good morning, students. Here, today we will continue with the same topic, that is the chromosomal theory of inheritance. Okay. Chromosomal theory of inheritance. Okay. So in this theory, as I told you, Mendel work. Okay. Mendel published his work on inheritance of genetics or inheritance of characters. Okay. So here Mendel published his work on what? On the inheritance of characters. Mendel published his work on inheritance of characters. How the characters are going to transmit from one generation to another generation. Okay, so he has taken the characters in the form of genes or alleles. Okay, in the year 1865. After that, but for several reasons, it remained unrecognized. Till what time? Till the 1900s. His work was unrecognized. Okay. After that, due to the firstly communication is there, due to which his work was unrecognized, and in those days, whatever the work he has published, first thing, communication was not good. Okay, this is the one reason due to which his work was not good at that days. So, due to which his work was unrecognized. This is first reason. Second reason, what he told, his concept of genes or factors. Second thing is the concept of genes. Genes, which are also known as the factors. What he told, the genes are factors in the Mendel's word. What he told, these are the discrete units that control the expression of the trait. What are these genes or factors? These are discrete units that control the expression of a trait. Okay, so this is the second concept. The third concept here. See here, what he told in the first concept, why his work was unrecognized. His work unrecognized. Till what time? Till the 1900. For what reason? This is the first reason, communication was not so good. This is the second reason, the concept of genes are factors. Okay, what are the genes? In other words, in Mendel words, what he told, genes as the factors. Okay, so these are the discrete units that controls the expression of the traits and of the pair of alleles which do not blend with each other. This was also not accepted. This concept is also not accepted. Not accepted. Okay, this is the second reason. Then Mendel's approach of using mathematics or mathematics to explain the biological phenomena, that is also disproved. That is the third concept. What is the third one? Third concept, Mendel's using mathematics in biological phenomena. Okay, what is that mathematics? What he used here means in monohybrid, dihybrid, test cross and all, he used a ratio, which is a mathematical expression. Okay, this also is proved or not accepted by other biologists. For these reasons, for these three reasons, the Mendel's work was unrecognized for till this time. Okay, so these are three concepts due to which the Mendel work was unrecognized. Then next, what happens in the year 1900? Some other scientists made some more efforts on his work. Okay, so what are that? Who are that other workers or other scientists who had put forth their efforts? So let's see that one. Next, here you see that in the year 1900, in 1900, there are three scientists whose name is D. Veris. Three scientists. Their name is first one D. Veris, Corals. Second is Corals, and third is that is one small. Okay, what are that? D. Veris, Corals, and one small. These are the three scientists who independently rediscovered the Mendel's result or Mendel's work on the inheritance of characters. So here, these three scientists, 1, 2, 3, they rediscovered the Mendel's inheritance of character. So here, rediscovered Mendel's result on Mendel's results on inheritance of character. Means once again they work on what? On the inheritance of character, which was unrecognized by whom? By the Mendel, by other people who Mendel has worked many times, but it was unrecognized. But these three scientists, once again, they rediscovered, here you see, rediscovered the same thing what Mendel has been done. Okay, so on what? On inheritance of character. So also by this time, due to advancement in microscopy, okay, so they used one thing here, that is a modern technique, that is they used, these three scientists, they used use of or advancement in microscopy. Advancement in microscopy. They used a particular instrument or a device that we are calling it as the microscope, that were taking place. Scientists were able to carefully observe the cell division by using the microscope. Here, either it may be a simple, compound, electron. Okay, so by using this advancement in microscopy, they are clearly known to, they are clearly visualize the what? Cell. Okay, cell in that what they visualize means carefully observe the cell division. See here by the use of microscopy, they carefully observe the cell division. What they observe? They observe the cell division. Okay, so here these were these whatever the during see here they observe the cell division where they observe in the cell where they observe the cell division means by the use of microscope they visualize the cell and that cell is going to divide. That also they had seen, observed. Okay, that division is going to be in that they saw observe the nucleus. In the cell, this is the cell. In that cell, they observe the nucleus. In that nucleus, they observed some structures. Okay, they observed some structures in the nucleus that appears to be double and divide just before each cell division. See here, in this nucleus, there were some structures, and that structures are double. First, they will get doubled. Then next, what they will do? First, they will get doubled. Then divide. Next, they will divide before the cell division. First, they will divide. Then the cell is going to divide. What is this structure? What are these structures means? These were the chromosomes. What are the structures in the nucleus? That is the chromosomes. Okay, first they will get doubled. The next, see, for example, this is the nucleus. Okay, in this nucleus, they observe some structures like this. Like this. Some structures they saw using the microscope. So what happens? These structures first they will get doubled like this. They will occur in a pair like this. First they will get doubled, then they will start to divide. Okay, this is doubled structure. Then they will start to divide in this way, like this. After that, the cell will start to divide. First, this division will take place. Okay, then next, the cell will start to divide. Like this cell division, they observe. Who are they? Vigorous, Corens, and von Tesma. And what they told me, so these were called the chromosomes. As I told you, the structures which are doubling and dividing, that structures are known as the chromosomes. What do you mean by this chromosomes? Means chroma means colored body. Here you see that structures are the chromosomes. Chroma means colored. Chroma means that is the color. Somes means that it is the bodies. Some colored body they observe. Okay, colored body as they are visualized. These chromosomes are visualized only by the use of a particular stains. Visualized by the use of stain. Stain means what? That some colors they have to use for observing that particular chromosomes in the cell. Okay, so they observe like this. So this is about the discovery of what? Chromosomes. Listen here. First, Mendel's work was unrecognized due to some concepts. Okay, after that, these were the scientists who rediscovered their results on what? On inheritance of characters. In that, by the use of microscope, they observed the cell division. Already you had studied about the cell division. Okay. They observe carefully what? That is the cell division. In that cell division, what they observe means first the nucleus is going to divide. That nucleus has some structures. Okay, that nucleus has some structures, and what are that structures? Means that structures are the colored bodies or the chromosomes. So they don't know the term chromosome. So something is going to seen there in the nucleus, which is first it is going to double, then they are going to divide. Like that we saw. Okay. And after that, what were their structures? That were the colored bodies. Some color is going to observe, and they have some structures. By using some stains and all, they recognized that particular structures in the nucleus were the chromosomes. Okay, so this is about what? About the chromosomes. Okay, the next in the year 1902, further what happens? These all things taking place in what? 1900. Next, what happens during the 1902? Let's see about that one. Next, in the year 1902, okay, here in the year 1902, two more scientists, their name was Walter Scruton and one more scientist, that it is Theodore Boveri. Theodore Boveri. These are the two scientists who brought Mendel's results on, brought a comparison between Mendel's conclusion about genes and their division of mitosis and as well as the meiosis. So let's see here, Walter Scruton and Theodore Boveri. These are the two scientists. What they did means brought a comparison between. They brought a comparison between about genes. Okay, they brought a conclusion between what? Between the genes and the behavior of chromosomes. Genes and behavior of chromosomes that is during mitosis and meiosis. Mitosis and meiosis. See, what these two scientists done means they brought a comparison between Mendel's conclusion about genes. What Mendel told? Mendel said that genes or the factors are the discrete units. which controls the expression of the trait. Okay, that conclusion he had given. And what they brought the comparison or conclusion between the one parent cell will give rise to the two new cell or the two daughter cell. When? During the mitosis. But this meiosis, which is also known as the reductional division, this meiosis is of two types, that is the meiosis 1 and second is meiosis 2. Here also, meiosis 1 is known as the reductional division, but this meiosis 2 is known as the equational division. Okay, in mitosis, one phase is there, only one phase. One parent cell will give rise to, see, always when a cell will undergo into the mitosis, they will give rise to only two cells. Okay, but when meiosis is there, for example, this is one cell which will undergo into the two phases of cell division. One is meiosis 1 and another is meiosis 2. First they will form, it's a reductional. Meiosis 1 is the reductional. So here they will form, reductional means here the chromosome number, it is going to reduce. So these cells will become NN. Then again these cells will undergo into the meiosis 2, which is the equational division. So here they will form 4 cells. This is similar to mitosis. Okay, but here there is a reduction is going to take place, that is the meiosis. So during this mitosis and during this meiosis, here I had written young. What do you mean by this young? Young is the one set of chromosome. One set of chromosome means if a cell is having one set of chromosome, then we can call it as the haploid cell. One set of chromosome means how many chromosomes? That is 23 chromosomes. Okay, here I have mentioned 2N. This is the diploid cell. It consists of 2N, that is diploid means 2 sets of chromosomes. Okay, 2 sets of chromosomes means that it is 46 chromosomes. Okay, so this all cell division is mainly dependent, or the cell division is some chromosomes are going to change over here. The number is going to change over here. In some division it is going to reduce, in some division it will be equally distributed to the new cells. Okay, so first of all, these two scientists, they brought a comparison between Mendel's conclusion about genes. About genes, what he told means, genes are the discrete units as they contains the information for a particular, or controls the expression for a particular character. Then behavior of chromosome. What is the behavior of chromosome during mitosis and meiosis? So I had cleared what do you mean by mitosis and what do you mean by meiosis. Okay. See here, one more thing they had told, that is the Sutton. Okay, what he told means, let's see. Here you see Sudan. Among the two scientists, one is Sudan. What he told? He told that an organism is having 1,000 of traits. See here. An organism has 1,000 of traits. Traits means characters. Height is there, weight is there, color is there, eye color is there, hair color is there. Okay, eyeball size is there. Many characters are there. You take one example of human, okay, which will have many characters. Thousands of traits. Traits means that is the characters. Whether for that 1,000 of characters, whether for each character there will be presence of one specific chromosome. For see here, there will be presence of 1,000 of traits, but not presence of 1,000 of chromosomes. Okay, here, but not thousands of chromosomes. Okay, so here, hence, so see, for one trait, this is one chromosome. Okay, this one chromosome is meant for what? Height. Whether this is correct? No. On this chromosome, there will be presence of genes. Okay, on these chromosomes, there will be presence of what? Genes. These genes, it may be many. Okay, these genes on a chromosome, it will be many in number. These genes are responsible for specific character. Okay, see here, genes are responsible for what? Specific trait or character. Genes which are present on chromosome, but not the chromosomes are responsible for each character in an organism. The chromosome will have the genes, or the genes will be present on the chromosome, which are going to present for a specific character. So here the idea of chromosome theory of inheritance was put forth in the year 1902. Okay, by Bovary and Sudan, and confirmed by whom? It was confirmed. This chromosomal theory, it was confirmed by one more scientist whose name is Morgan. What was his name? His name was the Morgan. In the year 1933. This theory was confirmed by whom? By the Morgan. According to this theory, the material for inheritance, according to this theory, what they told means the material for inheritance is genes. Okay, what Mendel told, genes are the discrete units. The same thing they are also telling, that according to this theory, material of inheritance is the genes. Okay, and these genes are present on the chromosome. Genes which are present on chromosome. This is one point. And this chromosome, it is that, so present on the chromosome, and it is the chromosome that segregate and sort independently. These chromosomes will segregate independently Whatever the chromosomes, they will get independently during meiosis. Segregate independently during what? No. During meiosis. That means the reductional division. These chromosomes are going to separate from each other. They are going to divide. Okay. So they are going to separate from each other during what? During meiosis. And next, and recombine. They will separate during meiosis and recombine during fertilization. Okay. This chromosome will recombine during fertilization. Okay, they will get separated during meiosis, but during fertilization, when the gametes are going to fuse, at that time they will recombine. Okay, so one comparison is there between the what we call that is the behavior of chromosomes and the genes. That table I will write here. See, whatever I had explained about the meiosis, they are going to separate, they are going to recombine, that I will explain through a diagram. See, in a meiosis, first phase, we had studied about the cell division or cell cycle, in which there will be two phases. First, see here, this is the cell cycle. G1 phase, S phase, then G2 phase. This you had studied. Okay, so here there are two chromosomes which are present on the same locus. That chromosomes we can call it as the homologous chromosomes. You see here, these are the two chromosomes and they are present in pairs, double. Okay, this is during what? During the G1 phase. During G2 phase, these chromosomes are going to double. Okay, how means? You see, in this sense, they are going to double. This is about the G2 phase. Here, this one I will shade for better understanding. Okay, these two chromosomes are present at one locus, and these two chromosomes are present at one locus. This type of chromosome, what we can call? We can call it as the homologous chromosomes. Okay, so this is also the shaded part. Okay, for better understanding, I am representing it with the different forms. Okay, so here during G1 phase they are single form. G2 phase they are going to double. You can see here 2 arms are there, means here 4 arms are there. Okay, it is going to double. But during anaphase, during metaphase they will come at the center. But during anaphase, what happens means they are going to separate. This is the meiosis, anaphase, and that too meiosis 1. Before that, they will segregate. This is the equatorial plate. During metaphase, they will come under the equatorial plate, but during anaphase, they will segregate from each other. Okay, the same thing you can see with these chromosomes also. Like this, they are going to move towards the opposite poles. Okay, they are moving towards the opposite poles. So this is about the anaphase 1 or the meiosis 1 anaphase. The next during the anaphase 2, what happens means meiosis 2, that means anaphase 2. Okay, after completion of meiosis 1, 2 cells are going to form. In that the chromosomes will be like that. But during the anaphase 2, they are going to segregate like this. In this way. Okay, they will segregate. Lastly, this will form the 2 chromosomes, and that we can call it as the. In this way, they will form the 2 chromosomes because they will segregate and they will distribute it to the 2 cells. Okay, here there is formation of 4 cells, which 4 chromosomes are going to distribute it here in this way. So this each one we can call it as the one set of chromosome will be present in one cell. So how many cells are there? 1, 2, 3, 4. 4 cells are going to form, and these cells we can call it as the germ cells or the gametes. During gamete formation, the chromosomes are going to segregate. Okay, that only we are going to explain in chromosomal theory of inheritance. In the next class, I will explain about same thing about the chromosomes and the behavior of chromosomes and genes during the mitosis and meiosis. Okay, thank you.